Hello students, in biobotany the first lesson is asexual and sexual reproduction in plants. In this lesson, today we are going to see first two topics, asexual reproduction and vegetative reproduction. What is reproduction? One of the essential features of all living things on the earth is reproduction. Reproduction is a vital process for the existence of a species, and it also brings suitable changes through variation in the offspring for their survival on earth. Plant reproduction is important not only for its own survival but also for the continuation and existence of all other organisms, since the latter directly or indirectly depends on plants. Reproduction also plays an important role in evolution. Milestones in plant embryology. 1824, G.B. Amici discovered the pollen tube. In 1848, Hofmeister described the structure of pollen tetrad. Professor P. Maheshwari was an eminent botanist who specialized in plant embryology, morphology, and anatomy. In 1934, he became the Fellow of Indian Academy of Science. He published the book titled An Introduction to the Embryology of Angiosperms in 1950. He established the International Society for Plant Morphologists in 1951. Now, reproduction. Two types of reproduction: asexual reproduction and sexual reproduction. In this, what is asexual reproduction? Without the involvement of gametes is referred to as asexual reproduction. Some of the methods include formation of conidia in Aspergillus and Penicillium. Budding takes place in yeast and hydra. Fragmentation in Spirogyra. Production of gemma in Mercantia. Regeneration in Planaria. And binary fission also takes place in lower species. The individual formed by this method is morphologically and genetically identical and are called clones. Next, vegetative reproduction. In this, two methods, natural method and artificial method. In natural methods, natural vegetative reproduction is a form of asexual reproduction in which a bud grows and develops into a plant. The buds may be formed in organs such as root, stem, and leaf. The unit of reproductive structure used in propagation is called reproductive propagules or diaspores. In this picture, some of the examples have given. Vegetative reproduction in root. Example, Murraya. underground stem and subaerial stem modifications, rhizome, gingiber officinale, in tuber solanum tuberosum, bulb in allium cepa, runner centilla asiatica, stolon fragaria, offset pistia, sucker chrysanthemum, bulbous dioscorea and agave. These are the few examples of vegetative reproduction the above picture have given for this. Vegetative reproduction in leaves. In some plants, adventitious buds are developed on their leaves. Example, bryophyllum, scilla, and begonia. In bryophyllum, the leaf is succulent and notched on its margin. Adventitious buds develop at these notches and are called epiphyllous buds. Now, advantages of natural vegetative reproduction. Only one parent is required for propagation. The new individual plants produced are genetically identical. In some plants, this enables to spread rapidly. Example, pinifex. Horticulturists and farmers utilize these organs of natural vegetative reproduction for cultivation and to harvest plants in large scale. Disadvantage of natural vegetative propagation is new plants produced have no genetic variation. The next method is artificial method. In this artificial method, there are two methods: conventional method, modern method. In conventional methods, there are three methods: cutting, grafting, layering. What is cutting? Depending upon the part used, it is called as root cutting, stem cutting, and leaf cutting. The next one is grafting. This picture have shown types of grafting: bud grafting, approach grafting, tongue grafting, crown grafting, wedge grafting. In this, the two portions used, the two parts of different plants are joined so that they continue to grow as one plant. Of the two plants, the plant which is in contact with the soil is called stalk, and the plant used for grafting is called scion. Example, citrus, mango, and apple. Types of grafting. Once again I tell you: bud grafting, approach grafting, tongue grafting, crown grafting, wedge grafting. The next one is layering. There are two types of layering: mound layering and air layering. Layering means the stem of a parent plant is allowed to develop roots while still intact. Example, ixora and jasminum. Types of layering: mound layering, air layering. Advantages of conventional methods. The plants are genetically uniform. Many plants produced quickly. Some plants produce little or no seeds. In others, the seeds produced do not germinate. In such cases, plants produce in a short period. Propagated more economically, by vegetative propagation. This is resistant and high yield can be grafted and grown as a new plant. Disadvantages of conventional methods. Use of virus infected plants produces viral infected new plants. Vegetative structures used for propagation are bulky and so they are difficult to handle and store. Now, modern method. The genetic ability of a plant cell to produce the entire plant under suitable conditions is said to be totipotency. The growth of plant tissue is special culture medium under suitable controlled condition is known as tissue culture. The regeneration of a whole plant from single cell, tissue or small pieces of vegetative structures through tissue culture is called micropropagation. These are the few methods take place in modern method. Now, advantages of modern methods. Plants multiplied rapidly in a short duration. Genetically identical plants grown in any season to produce many plants. Plants do not produce viable seeds and seeds that are difficult to germinate propagated by tissue culture. Endangered plants can be propagated by this method. Disease-free plants produced by meristem culture. Cells can be genetically modified and transformed using tissue culture. Disadvantages of modern method. Requires skilled workers for this method. Sterile condition adds to the cost. Clones are genetically susceptible to new diseases or changes in environmental conditions will wipe out the species. Sometimes callus undergoes genetical changes which are undesirable for commercial use. Now, that's all about this class. Now, home test. You write home test. Two-mark question. What is reproduction? And next question is, what is clone? And 5-mark question is, advantages of natural vegetative reproduction. Thank you. I once again repeat the homework. What is clone? 2-mark question. And what is reproduction? Five-mark question is advantages of natural vegetative reproduction. Thank you.